Hello, thank you for attending our AI for Drug Development tutorial. My name is Danica. I am the Director of Machine Learning at ACOE of IQVIA. IQVIA is one of the world's largest human data science company, and we are also the largest clinical research organization. We have lots of experience with clinical trials. It is my honor to be here and introduce some of our recent innovations on using deep learning for assisting clinical trials. The drug development process is mainly about clinical trials. As we all know, a drug needs to go through at least 3 trial phases before it can get FDA approval. These trials are labor-intensive and time-consuming, which often involve huge enrollment costs. Our AI methods are designed to alleviate such pain and reducing the trial costs, especially trial enrollment costs. In our work, we mainly focus on the following tasks that can be enabled by AI and real-world data. I listed them here, and there are 3 tasks. The first one is site or doctor selection that help target on clinical trial sites that are accessible by more patients, or finding doctors whose experience and networks make them better fit for conducting a new trial. The second task is called patient trial matching, which we aim at pinpoint the right patients for a particular trial and recommending them to enroll to the trials. And the third one is the ongoing work, which is called trial outcome prediction. We want to predict the trial outcome before a trial actually started. And here in this talk, I will mainly focus on the first 2 tasks, and I will briefly highlight our ongoing work and some future direction for the third task. For the first task, we will talk about our publication, Dr2Vec, which is published on AAAI 2020. This is in collaboration with our colleagues at IQVIA, Cedar, Lucas, Elizabeth, as well as our collaborator, Professor Jimon Song from UIUC. For this task, we would like to determine which doctors we should approach for conducting a new clinical trial for a particular disease. In the past, such a decision-making was performed by our specialists based on their knowledge and experience with the doctors they know. However, these are subjective and restricted by limited human knowledge and experience. We wonder if we could leverage the massive clinical data that we have about doctors, their patients, and the previous trials they help conduct. Perhaps we could derive better actionable insight that are more generalizable and that help us provide a better recommendation for doctors to trials. Therefore, we aim at develop an efficient representation learning method for doctors and their functions in the clinical trial ecosystem. From the data perspective, each doctor has seen a set of patients whose health conditions are encoded by longitudinal patient EHR. Also, the doctors have participated in a set of trials where the information are encoded by multiple types of trial features, including the unstructured eligibility criterion text and the categorical features that describe the clinical trials. And both the patient data set and the trial data set can help us understand the expertise and experience of a doctor, which further sheds light on whether the doctor could be a better fit for a new trial. However, there are 2 challenges we need to address in order to obtain a useful embedding. The first one is, how do we capture the time-evolving patterns of doctors and their experience or expertise? The second one is, how how do we learn a dynamic representation based on the corresponding child? Some doctors, their areas are quite general, so they can have multiple directions. When we learn the embedding, how do we focus on the more relevant direction rather and ignore the less relevant ones? To solve the two challenges, we proposed the Dr2Vec, which is a dynamic memory network-based embedding framework. Dr2Vec has 4 modules. The first 2 modules are patient embedding and child embedding modules. And the second one is the dynamic doctor memory network module. The last one is the prediction module. So let's describe these modules one by one. For the first 2 modules, the hierarchical patient embedding and the multimodal child embedding module, they actually perform the preprocessing step and take the raw data as their input and then encode the 2 entities, patients and trials, into a machine-readable vectors to be used in the dynamic memory network. For the patient data, they are longitudinal EHR data. They are like a sequence of medical codes. And we pass them through a bidirectional LSTM, and then they become the vector which used as the memory for the memory network. And for the clinical trial data, they come with 2 modalities. One is the free text, the unstructured text. We encode them using BERT. And the second one is the categorical features for clinical trials. Because they are low dimensional, we simply encode them using the MLP and concatenate the 2 parts to become the query for memory network. And now comes to the main module for Dr2Vec, which is the dynamic doctor memory network. The Dr2Vec memory network takes 2 embedding as its input. One is the patient embedding. The other one is the trial embedding. And the patient embedding serves as the memory of the dynamic memory network. And the trial embedding is used as the query to retrieve relevant dynamic memory. And we just learned the patient embedding, and we also know that a doctor has seen a set of patients. So these patients can become, the patient embedding can be aggregated to become a doctor embedding. For a particular doctor, the embedding for all his or her patients will be stored as a dynamic memory. One patient a row, the columns are all the feature dimensions. When we make a prediction to learn the fitness that a doctor is for a new trial, we use the embedding for that trial, the trial embedding of the query, to retrieve the relevant part of the memory, and finally output the dynamic embedding for that doctor with respect to that trial. And here, the relevance is determined by an attention mechanism. And the output embedding will be concatenated with another doctor embedding that encodes the static information of a doctor. And then the final embedding will be used to make prediction. And the outcome will be a number that is predicted enrollment rate between 0 and 1. And based on this number, we can cast the task as a simple regression task, because the prediction outcome is a number between 0 and 1, and it indicates the enrollment rate. Or we can cast the task as a classification task, which we simply perform binning of the prediction results. And we tested our method using data collected from both public websites, such as clinicaltrials.org and our proprietary clinical data. For the classification task, the performance is measured by PRAOC, which is a good metric for skill data setting, because most of the enrollment rate are in the middle. It's not super high or super low. That the mode is in the middle, around 0.5 or 0.6. And the regression is measured by R-square, which is the square of the correlation between predicted scores and actual scores. For both metrics, the higher the better. And we test them on trials formed during 2014 and 2019 across 28 countries. And for the patient data, we also use the IQVIA claims data. We compare Dr. Tuvek with the following baseline approaches, including the median, which is used in practice. We simply consider the median enrollment rate for each therapeutic area as the estimated rate for all trials in that area. We then compare with a few powerful shallow models, which includes logistic regression, random forest, and ADA boost. We also compare it with a few deep neural network, general deep neural network, including MLP and LSTM. Also, we compare it with a previous state-of-the-art model in this particular task, which is called a deep match. This is also a work published by colleagues from IQVIA. From the experimental results, we can see that Dr. Tuvek outperforms all baselines. Among the baselines, all models that capture temporal patterns outperform the ones that do not. We also tested the performance on two common scenarios. One is that we train a model using historical data from the US and test it on some countries that are less populated or newly explored. And the other setting is we train a model on trials that are about common diseases and test it on trials about rare diseases. Both settings can evaluate the generalization power of a model, and results indicate that Dr. Tuvek works much better than the baselines. So we have a lot of result discussion and analysis in our paper. If you want to learn more, feel free to take a look at it. Next, I'm going to talk about how to perform doctor or site selection for epidemic disease or pandemic diseases, such as the site for testing COVID-19 drugs or vaccines. We recently published two papers on this topic. One is on JAMIA, and the other one is in forthcoming AAAI 2021. And today I will briefly discuss the JAMIA paper, the STEM model. For those who are interested in the second paper, our paper can be found online. You can feel free to take a look. And what's special about this type of task? To perform site selection for epidemic diseases or pandemic diseases, such as flu or COVID, we are facing new challenges. Challenges come from lack of data or effective modeling tool. So data-wise, since the pandemic diseases are often seasonal, acute, and infectious, longitudinal patient health records do not encode so much useful information or patterns for them. Model-wise or tool-wise, the existing models that we use for modeling epidemic and pandemic diseases are the compartment model, such as the SIR, the susceptible, infectious, and recovered SIR model. They are mainly designed to model how public health interventions may affect the outcome of a disease. They are not designed for accurate prediction. And if we want to decide which location and which site we should build a new clinical trial to enroll patients for testing COVID-19 drugs or COVID-19 vaccines, then we really need accurate prediction for how many people will be affected in that region. So, motivated by this real-world need and challenges, we propose this STEM model, which we will introduce now. So the STEM model is called a Spatial Temporal Attention Network. It is for pandemic prediction. And since these diseases are often infectious, we model each location, which could be a city, a county, or a state. We model each location as a subject, and we construct a location graph to connect these locations. We describe each location using many node features, and they include the daily counts for diseases, related diagnosis and procedure codes, as well as other demographics information. We listed a few here. You can find the full list in our paper. With this up-to-date information, it is likely to capture the trend and the momentum of a disease outbreak, and thus more accurately predict the number of potential volume of trial participants in that location. We then connect these nodes using geographical similarities between the nodes. The edges can also incorporate transportation volume to better capture the disease transmission. And the graph is then fed into a graph attention network to extract spatial temporal features and learn the graph embedding for a target location. Next, the graph embedding will be fed into a GRU to extract a temporal relationship. The hidden state of GRU will be used to predict future number of infected and recovered cases. Unlike the existing time series-based model, which can only predict the outcome for next time step, we can predict a p-step infected and recovered cases. So this can be, we tested, we can effectively predict up to 20 days for the future. For the STEM model, we also use an additional transmission dynamics loss based pandemic transmission dynamics to optimize the model. We tested the model on our real-world data, and our STEM model outperforms traditional epidemiology models such as SIR, SEIR, and some deep learning models on both long-term and short-term prediction, achieving up to 87% reduction in mean squared error compared with the best baseline. We also tested to predict the future 20 days. Our model has been used to show less overfitting and perform much better results than the SIR and SEIR, and it has been used in support of many COVID-related trials within our company, and often show better performance. Okay, now we are ready to talk about the second task for AI for drug development, which the second task is patient trial matching. After doctor and site selection, we now need to pinpoint the right patient to recommend to a trial. We name this task patient trial matching. We published 2 papers in 2020. Our first generation of patient trial matching model was published in WWW 2020, and the model that is currently in use is the second generation model called Compose, which is published in KDD last year. And this is also in collaboration with our colleague Lucas and our collaborators Junyi and Jimeng from UIUC. And today I will focus on talking about the composed model. If you are interested to know more background, you can feel free to check our deep enroll paper as well. Okay, so our composed model is about matching patients to trials. And this is aimed at solving the patient enrollment challenge. As we all know, the patient recruitment is a long-standing pain. The recruitment cost is very high and estimated to be around $6,000 to $75,000 US per patient. And this raises critical and also recruitment failure very common. Statistics shows that 50% of trial delayed and 25% cancer trial will fail, all due to enrollment problem. And this raises a critical question for us to answer. Do we have more effective and efficient way to help patients find trials, or help trials or doctors identify patients who are fit for a particular trial? Especially given that we now have access to many types of clinical trial related data, as well as advanced deep learning, machine learning technology. There are two types of trial data we can use here. One is the patient EHR, which can be seen as a type of high dimension sequence data, and it provides essential health information for patients. The EHR data are sequences of diagnosis, medication, and procedure codes. The other type of data is the trial eligibility criterion, or trial EC, which includes both inclusion and exclusion criteria. They are written in free text. Our task is essentially a cross-modality matching task that match a sequential data to a piece of unstructured text. However, the application has its own challenge. It's unlike a general cross-modality matching task. The first challenge is how to deal with multi-granularity medical concept. The unstructured ECs often encode more general disease concepts, while the structured patient EHR often represent patient conditions using more specific medical codes. For example, a patient with pleural cardio adhesion on EHR data can be recruited to a trial that requires more general cardiovascular diseases. And there are more diseases that belong to this category. So it is non-trivial to match medical concepts with heterogeneous granularity in different data modality. The second challenge is how to deal with the many-to-many relationship between patients and trials. In practice, each patient may enroll more than one trial, sometimes, and vice versa. To conduct matching across different modalities, we need to learn patient and trial embedding in the same embedding space. Since each trial generally focuses on certain diseases, the semantic distance between the trial and criterion may be large. However, all existing works align the patient embedding to different trial embeddings, which may confuse the embedding function and lead to inferior matching results. And the third challenge is how to handle inclusion-exclusion criterion. The trial criterion comprise inclusion and exclusion criterion. They describe what are desired and unwanted from a target trial. The same sentence may have completely different meanings and lead to different patient cohorts depending on which category it belongs to. So we really need to explicitly handle the two type of criteria. With all three challenges to solve, we propose Compose, which is a pseudo-Siamese network consists of two branches, a convolutional neural network-based branch to learn the trial eligibility criterion embedding, and a taxonomy-guided memory network branch to learn the embeddings for patient EHR. Then we use an dynamic alignment and matching module to generate final output. Compose will embed two modalities of data from ECs and EHRs into a shared latent space. In particular, we use the EC embeddings as a query to read memories in the memory network. Finally, we use a cross-entropy classification loss and a composite similarity loss term to jointly predict matching scores. Let's talk about more detail for how the Compose learns embedding and make predictions. First, we learn the trial eligibility criterion embedding using convolutional highway network. We hope to learn the EC embedding can retain sentence-level semantics while also capture important detailed information in sentences. To this end, we first use the pre-trained BERT to generate contextualized word embedding for each word in the eligibility criterion. Specifically, we apply the clinical BERT pre-trained on PubMed text and Mimic 3 doctor notes. Then, we use a multiple one-dimensional convolutional layers with different kernel sizes to capture semantics at different granularity level. Next, we feed it into a convolutional highway layer, which has been widely used in many NLP tasks to extract text semantics. Finally, we use the max overtime pooling to reduce the dimension and obtain the final embedding for the EC sentence in current trial. Each dimension in the embedding denotes a semantic feature captured by convolutional filters. Next, we learn the taxonomy-guided patient EHR embedding. Given a patient's EHR data, each visit can be represented by three types of medical concepts: the diagnosis, medications, and procedures. Each type of medical concept can be divided into different levels, from a more general concept to a more specific one. In this work, we use the Uniform System of Classification, the USC taxonomy, to divide each concept into 4 levels. To this end, our patient embedding memory network consists of 3 separate memory networks to store diagnosis, medication, and procedures respectively. Also, another slot to store demographics information for patients. Then each memory network has 4 memory slots to store information from the fine-grained to coarse level. Then, in clinical trial ECs, medical concepts are described in natural language rather than medical concepts. Therefore, using the text description of medical codes to embed patient's EHR, we, instead of using one-hot encoded codes, can help the model find a matching concept. Here, we use the BERT with max pooling to generate the concept embedding. Then, because the patient's EHR are sequential data, we update the memory slots at each visit. We adopt the erase followed by add update mechanism as this equation. Then at each time step, the memory in each slot will be updated using this equation. Next step, we will conduct attentional record, alignment, and dynamic criteria matching. A previous patient trial matching works learn an alignment matrix which map patient's EHR embedding and EC embeddings to the same latent space. Trying to push the patient EHR embedding to be close to different EC embedding may confuse the embedding function. Or the patient embedding could be mapped to near average position of these EC embeddings. To overcome this problem, we let each EC correspond to the sub-memories that match current trial best. Concretely, we use the inclusion criterion and the exclusion criterion embedding as queries to attentively visit the memory network. We compute the attention weight for different slots as this equation. Then we obtain the best matching memories by taking the average sum of all the memory slots. Last, to explicitly handle the exclusion and inclusion criterion to solve our third challenge, we design a loss function that consists of two parts. We first use a cross-entropy loss term to optimize the classification performance between the predicted matching results and the ground truth. In addition, we also use a loss term to explicitly handle the matching between patient embedding and the EC embedding for inclusion criterion and the exclusion criterion. This boils down to minimize the distance between the retrieved patient memory and the embedding for inclusion criterion while maximize the distance between the memory and the embedding for exclusion criterion. In this equation, if a patient matches the inclusion criterion, the model will minimize the cosi distance between the two embedding to make the term close to zero. If the patient is excluded by an exclusion criterion, the distance between the two embeddings should be no more than n alpha. The loss term enables the model to extract different features in the inclusion-exclusion criterion and then help decide which to include or exclude to the patients. So now we finish describing the framework, and we tested the composed model on real-world data. We tested the matching performance across 590 clinical trials. For criterion level matching and trial level matching, our composed model shows significantly better performance than the previous state-of-the-art, which is also proposed by us, the deep enroll model. We also analyze its performance on different trial phases and across different diseases, and you can also find more analysis in our paper. What we want to highlight here is, in order to show how the attentional record alignment mechanism works, we have some case studies in our paper. Here we choose a trial which treats tumors. Compose will successfully matches this trial while all the baseline fail. Let's take a look. Why is that? For this trial, each criterion focuses on a single diagnosis or a medication. So it is difficult for baseline models to match each criterion to a longer patient record. However, the attentional record alignment mechanism in Compose in our model helps each criterion match the most related memory slots and therefore achieves dynamic matching. For example, the fourth criterion here, the pregnant or breastfeeding, is aligned to the demographic slot. And the second criterion here, receiving warfarin or other coumarin derivatives, is aligned to the LV2 and LV4 of the level 2 and level 4 of medication slot. since warfarin corresponding to a detailed medication, while the other one corresponding to a higher level category. So we have this dynamic matching and alignment really helps in finding right match, make a right matching. We also found some trials difficult to find matching patients. For example, here is a failure case. For example, for this early stage non-small cell lung cancer trial, all models achieve lower than 50% accuracy score, including ours. We selected some ECs in this trial to explore why. Our model will classify the 3 ECs to unknown because for this criterion, the EHR data is not enough, not sufficient to determine whether a patient will match these criterion as listed on these slides. And the trial recruiters need more data, need more tests to label this data and to identify patient cohorts. Okay, so here ends our second task. And for the third task, this is a new direction we are exploring. We have a paper in which called HINT, Hierarchical Interaction Network for Trial Outcome Prediction, leveraging web data. This is in submission. And the challenge we want to solve for the trial outcome prediction task is the clinical trials, they face uncertain outcomes due to issues like safety, efficacy, or problems with patient recruitment. If we were better at predicting the results of clinical trials, we could avoid having to run trials that will inevitably fail. More resources could be devoted to trials that are likely to succeed. So this is a very tempting outcome. In our most recent work, however, so for so many years we don't have this aggregated data resources for researchers to explore this task. In our most recent work, we proposed an hierarchical interaction network for a more general clinical trial outcome prediction for all diseases based on a comprehensive and diverse set of web data, including molecular information of the drugs, targets and disease information, trial protocols, and biomedical knowledges. Along with this paper, we will release the first of its kind trial outcome prediction data set, which we perform a lot of human labeling from clinicaltrials.gov, and we will release both a manuscript and data set very soon. So hope more people will join us to using machine learning and deep learning to improve the drug development process. Here are all the reference papers in my sections, and feel free to reach out to me. Here is my email, and if you have any question, feel free to shoot me an email. Thank you.